Could you please check?
One minute, one.
Yeah, there is he.
Good evening, Dr. Ankur Sharma.
Hello, Dr. Ankur ji. Namaste. Good evening.
Namaskar, ji, namaskar.
So, should I share my screen?
Yes, please, you can. Within 3 minutes, I will start.
Yeah, sure. I'm just trying to see if everything is visible or not. Are you able to see my screen?
Yes, Dr.
Yes,
Shah,
sir.
yes.
Okay, good. That's good. Let me know when you want me to start. I will be right on.
Just within 2 to 3 minutes.
Yeah, sure.
Actually, near about 200 participants have joined here, and 200 plus participants also joined in YouTube. So
Oh,
total, I think 400 plus participants.
that's very good. Incredible. Well, this is a very good workshop, and very happy that you have been conducting this for so many days. That's a big deal, and it's a very fascinating workshop.
Thank you so much, Dr. Shah.
Welcome.
आप लोगों के सपोर्ट से ही पॉसिबल है ये, पर्सनल नहीं कर सकते.
Right.
Sir, almost 200 participants are there. Shall we start?
Yeah,
Yes,
sure.
please, Nandini. Yes, please, Nandini. You can start.
Okay, sir. Am I visible and audible as well?
Yes.
Okay. So, a very warm good evening to everyone. As we continue to our journey in Winter School Bharat 2025, I welcome you all to the day 27 of this academic enriching program. I'm Nandini Sharma from AgriMeet Foundation Bharat, as you all know me. And this winter school has brought together some of the finest minds in Indian agriculture and sciences for the past several days. We have been learning from visionary scientists, understanding emerging technologies, and exploring future ready solutions that can strengthen India's agriculture resilience. Every session is designed to empower young researchers, academicians, and professionals with cutting edge knowledge, and today's session continues that tradition. It is my honor to welcome Dr. Anirban Guha, faculty member at the School of Biology, IISER Thiruvananthapuram, and one of the India's leading young plant biologists working at the interface of plant physiology, climate change, and ecosystem sustainability. Dr. Guha's research explores some of the most critical and timely questions of our generation. How do plants respond to climate change, warming, drought, hypersalinity, and extreme events? How do physiological and hydraulic constraints shape plant growth and productivity? What hydraulic targets can empower the yield and resilience of important industrial crops? By integrating ecophysiology, plant anatomy, metabolomics, he bridges plant structure with ecosystem function, providing powerful insight into how plants survive, adapt, and thrive under environmental stress. His work is vital not only for scientific advancement but also for ensuring long term agricultural sustainability in a rapidly changing climate. Sir, we are honored to have you with us today at Winter School Bharat 2025. Your expertise, your interdisciplinary approach, and your commitment to understanding plant resilience will greatly enrich our participants' learning experience. On the behalf of organizing committee, AgriMeet Foundation Bharat, UGC MMTTC Jodhpur, Faculty, research, and all participants, we extend a heartfelt welcome to you, Dr. Anirban Guha sir. We look forward for your enlightening session. We are very much grateful for your presence. Over to you. The session is yours, sir.
Well, thanks, Nandini, for your very kind introduction. Is my voice audible clearly?
Yes, sir.
Okay, good, because I had some throat issue because of the climate change. So maybe my voice is a little bit, cranky sometime. So anyway, my wholehearted congratulations to all the participants and the organizers for this wonderful workshop, which is really fascinating. And this is very timely, actually. And we all need this kind of workshop more and more. And we want young minds to come together and discuss and think and work to pursue uh, for whatever is needed for the sake of time and for the sake of our Indian, farmers, Indian people, and for the country overall. So today I decided to talk about a little bit on crop photosynthesis in the context of warming. And very quickly, I will say a few things about my institute. I am from IISER Thiruvananthapuram. This is in the southern tip of India. It is in Kerala, around 40-kilometer drive from the airport. And it is in the southern tip of the Western Ghat mountain. As you can see, our campus is just in the middle of the forest. This is an evergreen rainforest. It's one of the beautiful campuses of India for higher academics. And this is our School of Biology. Again, we have not only just biology, different schools we have in our institute, and some of our schools have physics, a school of physics, chemistry, maths, earth sciences, and data sciences. And we have also a couple of state-of-the-art facilities like high performance computing, Camry, etc. So I have been working on plants for last couple of years, and I have touched different kind of systems because as a scientist, I don't want to stick to a single system because I feel that because so much of plant biodiversity is there, and it will be really fascinating to know at least some of them. So I've worked on different kind of cereals models, different kind of horticultural models, different kind of tree species, natural forest tree species also. But today we will mostly talk about crops, some of the important cereals and oilseed crops, and also some of the horticultural crops. So we all know that the earth has been warming, and we all know why the earth has been warming. So the reason is that the earth is warming because of the accumulation of the CO2 in the atmosphere, carbon dioxide in the atmosphere. And this graph, if you look into the upper side of the upper panel, there is this blue curves, which shows that how the CO2 has increased from the last 800,000 years to the current time, from 240 ppm to right now it is 427. And if you look into this curve, you will see there are ups and downs, and see the lower panel, the red color, which is actually showing the temperature of the earth. So if you see that whenever the CO2 level on earth has dropped, the temperature also have dropped. Whenever it has risen up, the temperature has also risen up. So it clearly shows that the accumulation of CO2 and the increase in the global temperature is very much tightly correlated. But the problem is, if you see that the earth has not really seen this kind of high CO2 in the past history of earth. Right now, the CO2 concentration is really havoc. It has crossed 400. And because of that, all this kind of global warming scenarios are coming up. And this is a kind of simulated study which shows that if the temperature on earth is increasing at a certain degree, like from 1.5 degree to 4 degree, then there will be an uneven warming in the earth. Some of the part of the earth will warm more, but overall there will be a mean temperature rise. As you can see in the extreme right, where the earth looks like very much reddish, there you will see that some of the polar sides are more reddish compared to the central part. So it shows that some of the places on the earth will be getting more affected by the high temperature. Now, having a high temperature due to CO2 accumulation, how does it affect plants or our crops? It affects plants on two way, because CO2 is a greenhouse gas and it actually affecting, it is creating the temperature. So there is a direct effect of CO2 that it's causing the high temperature, which is affecting the photosynthesis of the plants. And also there is another effect of the plant, which is an indirect effect. So CO2 has this two kind of effect. CO2 itself, high CO2 is a substrate for the plant. When there is a high CO2, it is affecting the photosynthesis because the plants are getting more substrate. Whenever there is a high CO2, there is also increase in the temperature. So the plant systems are also sensing this high temperature. Now plant is this two signal, high CO2 as well as high temperature. Now, we really don't know very much clearly in respect to many plants that how these two signals, our plants, our crop plants, our native trees are going to sense and respond. That's why there are a lot of research. But there are very simple and very fundamental mechanistic understanding that we really know is that we know that when the temperature rises, the photosynthetic curve, that is the green color, it actually rises. So photosynthesis rises as a response to temperature, and it hits a tipping point, which is the center part of that bell-shaped curve. And then it drops because the photosynthetic processes are affected at a higher temperature. In contrary, probably the respiration, which means the energy releasing process in the plants, they will ramp up. So now this two important. One is the carbon gain process through which the plants are acquiring food, and the carbon loss process through which plants are breaking down the energy for its growth and reproduction. These two things will be working in tandem. And now highest temperature, in the high temperature, these two process, we need to watch and see that which processes are actually responding more, comparatively more in the high CO2. Now, as I was telling, that photosynthesis is very critical. We all know, because from the school, high school and colleges and so on, we have heard about photosynthesis. We have heard so much that when I teach at ISET, because I also teach heavily here. So sometimes the students say, "Oh, sir, it is so boring. Why you even talking about the photosynthesis?" But you see, the photosynthesis is such a mechanism, such a dominant mechanism on Earth, that we cannot really we cannot really let this go, because without photosynthesis, there are no crops, there are no sustainability, there are no life on Earth. But then what is photosynthesis, where it occurs? Actually, if you look into this, photosynthesis is deep down inside in the plant. If you keep zooming in into the leaf, you will see at some point of time here, all these globular structures are there inside the chloroplast. This is like button-like structure. When you cut it open, you will see these membranes are exposed. These are all thylakoid membranes. And inside this, all this fascinating machineries which nature and evolutionary process has created. So all these photosynthetic machineries are deep down inside, and these are not just one machinery, several machineries are there in and around this membrane. All this put together in tandem, when they work, the process, what they achieve is known as photosynthesis. But to keep it simple, it is the process by which the energy is captured by the green plants, which is the light reaction. And after the energy is captured, the energy is used to synthesize reduced carbon compounds from the carbon dioxide and water, which is a dark reaction. So basically, photosynthesis are a combination of these two reactions, light and the dark. But real life photosynthesis is actually several processes. And why we have to know all these processes? Because each and every processes are not stand alone. They are connected in a series, as a chain it is happening. But I won't go deeper, but just look into all these red boxes very quickly. You will see the processes: photophysics, photochemistry, photobiochemistry, and physiology. So this is the level of complexity. This is again another way to look into the level of complexity. Several reactions, several physical and biochemical reactions are happening. Coming to the C4 plants, we will start the discussion with the C4 plants. because C4 plants are really a very tiny part of all the crop systems what we have. Only 3% of the plant species are C4. Yet, fascinatingly, 30% of the world cereal production comes for the C4 photosynthesis. So, this is the wonder of the C4 plants. Very little, but then they have their impact on our, daily livelihood is tremendous. And I will not go deeper, but I will just quickly say that there are biological difference between C3 and C4. The C4 has a special mechanism. It has a mesophyll cell which is enriched with PEP carboxylase, where the CO2 is trapped. And this enzyme is unique to the C4. And then those CO2 is pumped inside to the Kelvin cycle. So unlike C3, which has just the Rubisco, C4 has two additional enzymes, PEP and Kelvin. And you better look into your, study books to understand how this extra step, this having a PEP actually helping C4 plants. I think most of you know, but if somebody does not know, please take a look into. Now, if you see what is the advantage of the C4 plants compared to the C3, you have to think in the crop perspective. Now, this C3 has a very lower, light saturation point. If you see this, this blue line, okay, which is the solid line. This is a C3 plant. Whereas you see this dotted line, it has a C4. And if you see that the C3, C4 plants have a very higher photosynthetic rate at a given light intensity. Now look into the temperature perspective. This y-axis is temperature. And if we look into the same responses, the C4 plant will have a high photosynthesis at a higher temperature compared to the C3 plants. So C3 plants looks like, God has made for the sake of, for the sake of tropical and subtropical areas where temperature is high, light is abundant. C4 probably will do much good. And that's why a lot of C4 crops are, coming into limelight. But interestingly, if you see what is here, I'm trying to establish a fact that even though they are working better at a high light and as a high temperature, their photosynthetic efficiency is really not great. If you look into how much of light out of 100% sunlight, how much of it can really convert into biomass energy, the C3 will have 4.6% energy conversion efficiency, whereas C4 will have 6%. So C4 is just 1.4% higher or better in terms of energy conversion. So it is really not that they're really great in photosynthetic efficiency. Now, if you look into this plant, the C4 grasses, these are basically grass, tall grass, erect structure. And you will see that they have these two important leaves, the flag leaf and the second leaf. These are the leaves which see the extremes, which means they see high light, they see high temperature, they see high diurnal fluctuations. But they also provide a lot of photosynthetes, assimilates to the grains when they mature. So these two leaves are very much important for these plants. Now, can the C4 photosynthesis be improved further? Because if we want to think about the production of the C4 crops, can we think that if the C4 photosynthesis can be improved, probably there is any scope or any chances whether the C4 photosynthesis can be still improved? Because these two leaves are really important. Their photosynthetic efficiency will be really important for the performance and for the yield. Now, one interesting aspect would be to look into the genetic variation, existing genetic variation, like whether there are certain genotypes already existing which have a better efficiency. Another aspect would be to look into this photosynthetic traits, whether they can really adjust and acclimate to challenging situation, because in the climate change, global warming, we are predicting that more and more challenging situations will come. So this photosynthesis, these photosynthetic traits, can it adjust, can it acclimate, can it cope up with the changing condition? Those things probably, if we try to find out, then we have some, tools. We can probably come up to some tools which we can use it. I will show some study from model system sorghum. And if you see this slide, here I'm showing two leaves. The green color indicates the flag leaf, the red color indicates the second leaf. Second leaf means the second from the top. And if you see, I'm showing the two responses here. On the left hand side, you have the photosystem yield. On the right hand side, you have the electron transport rate as a response to increasing light. What happens when you increase light on the leaves? The photochemical capacity of the leaf actually slowly it drops down because the system gets reduced. But at the same time, the system's electron transport capacity increases, which means as a response to light, the electron transport capacity of these leaves are continuously increasing. And you will be very interested, I think, with this observation that if you see the top leaf, the flag leaf, they have a very high electron transport capacity compared to the second leaf. So this is an interesting phenotype which is already there within the plant leaf profile. Now, if you see this phenotype, this profile across other genotypes, you will see that yes, this is actually holding true in this kind of C4 sorghum, where the flag leaf is actually having a high photochemical capacity than the second leaf. But interestingly, if you see, there is a very interesting genotypic variability. Some of the genotypes have an ETR of 44, some of them have ETR of 60, some of them have ETR of 66. So there is already a very interesting variability is there in terms of electron transport rate. Now, these are important traits which one can really look into if they want to improve this crop species. That will be really interesting. Now, I was telling you the acclimation, like, okay, these traits are good, these traits are there. Some genotypes have a better traits, which we can use. But can these traits, succeed in the face of climate change? For an example, if you see this graph, I'm showing that, yeah, compared to C3, C4 plants, they can do better at a high temperature. That's we all know. But then what we did is we did some interesting experiments. We ramped up the temperature over, couple of weeks. The first week we had a moderate temperature in the greenhouse, and the second week we cranked it up. We made the temperature very high by tweaking some of the greenhouse settings. And the third week we got back the temperature a bit down. And we wanted to understand how this photosynthetic traits in the leaves, in the crop, C4 crop, they're behaving, they're responding. And if you see, look into this right-hand side, the ETR profile, you will see that this black line, which is the response in the control condition, like the very low temperature condition, it was ETR was 65, whereas on the second week when the temperature really went up, the ETR actually responded positively and it went to 122, almost double. And the third week when the temperature was brought down, the ETR again got back. So this was the response in the flag leaf. So flag leaf actually, in this particular genotype, the flag leaf actually have a positive, trigger to the rising temperature. In the second leaf, the leaf which is just below the flag leaf, if you see, the same response was there, but the magnitude was less. Here, the magnitude was not this high, it was only 108. It was only 108, electron transport was only 108. Also, when it dropped, it also had a moderate drop. So you see that within the same genotype, the flag leaf and the second leaf, they are having a very different response to the temperature. This is very interesting because this could give you very interesting synthetic biology targets to, tweak the plant, leaf behavior, leaf, all the genes and proteins, and to make some leaves more productive under high temperature. Now, this profile we were able to establish in many other, genotypes, but you will see that there is a very prominent, there are three genotypes are here, 1, 2, 3. I'm showing their flag leaf and the second leaf in the bottom panel. And you see that already you can visibly, I'm not no need to explain that there is a huge difference in the electron transport efficiency genetically as well as as a response to temperature. So this is a quick view of what we have been doing in our greenhouse. This is my student Gauri, and she is measuring all this, whatever data I have shown. She's measuring, she is working hard, and she has been measuring. Okay, good. Okay. Wait, huh? I think it got a little blocked. Hmm. Okay. So am I still audible, Nandini?
Yes, sir. Yes, sir.
Okay, good. So now we have been talking about this crops, the semi-arid sorghum crops, what all it can have, traits, hidden traits. And now we will talk a little bit more about this photosynthetic machineries. You know, this machineries under high temperature, when they're performing better, what it needs. You know, when you have to perform better at a high temperature, you need to be tolerant. Without the tolerance, you cannot perform. Now, what is actually the tolerance or the resilience of the photosynthetic machineries? It is actually all this, you know, it is the machineries are made up of protein lipid structure. And these protein lipid structures are essentially thermolabile. Now, whether these structures will really be tolerant under high temperature, and there are certain genotypes which are intrinsically very tolerant compared to other, those kind of informations will be really important. Now, we are talking about high temperature, but really how high, how much high it goes. You see here some data which was done in a very different condition by tweaking certain interesting things. We checked the leaf temperature, which is in red, with the air temperature, which is in the gray, across different genotypes of the sorghum. And we will see that the leaf and air temperature did not vary. But under the condition where we did this experiment, there we did interesting changes where we removed, we actually increased the wind speed on the plant. We wanted to create a condition where there are a lot of winds will be flowing over the leaf. So when a lot of wind is flowing over the leaf, that time because of the high wind flow, the convective cooling of the leaves are really good, and that time the leaves are able to maintain a very good temperature along on par with the air temperature. But this is actually not a real life scenario. And real life scenario, when you see this kind of responses, you will see that sometimes the leaves actually warm up very higher than the air temperature. This is not a sorghum leaf. This is a leaf from other important plant species, but the message is same. You will see that different genotypes, they are warming up at a different rate compared to the air temperature. So whenever I'm playing a video, it is getting a little bit of hanging. The slide is taking some time to move. So what I will do is, I think this was the last video. Okay, so now you see this thing here. What I did is, here we did an experiment where we have increased the temperature of the leaf suddenly. And this black line is the air temperature. Now, when we increase the temperature suddenly, within 0 to 600 second time, sorry, so within
Yep.
0 to 600 second time, if you see that the flag leaf and the second leaf, they have a very different kind of temperature rise. Compared to the air temperature, there is a 5 degrees Celsius rise in the flag leaf. So your flag leaf, the crop, the first top leaf, which is so important, that is actually shooting up the temperature so rise. And the second leaf is also around 2 degrees higher. So this is a real life scenario where the leaf temperature will not equilibrate with their temperature and the leaf will be much hotter and warmer. And this behavior we called a megathermy behavior. And this behavior is not restricted to one crop species. Many important crop species we have tested and we have seen that, but there are variations crop to crop, genotype to genotype, variety to variety variations are there. So when the temperature of the leaf is shooting up so high, it actually becomes very important to really understand the tolerance of these photosynthetic structures. As I was telling you, this photosynthetic machinery is a thermolabile. Now, how we understood, we did a photosynthetic thermal assay to really identify genotypes or varieties which have a very high temperature tolerance. We used very interesting chlorophyll fluorescence techniques. These techniques are already well established. It was actually launched by the plant ecologist and then taken from them, then taken over by some of the crop physiologists. So here basically we heat treat the leaf or the discs and we check the signals when the leaf is actually doing good. But again, when we heat stress at a high temperature, the structure disintegrate. As I was telling you, these structures are not very thermotolerant. And then the signal, again, the signal, we tap the signal changes. Using these signals, we calculate different kind of tolerance markers like T50 and T critical. And if you see just within the sorghum is already acclimated to high temperature. But if you see just within around 14 genotypes, we were able to establish 2 degrees Celsius variation in thermotolerance. 2 degrees Celsius variation in thermotolerance is actually a lot. But this is another interesting thing. Is you see this, my student Amina is standing here holding a sorghum plant, which have varied with all kind of leaf numbering I have done from the top to the bottom. And if you see this heat tolerance capacity of the leaves, how it changes across the profile. If you see the first leaves have not much tolerance, then leaf 4, leaf 5, they are more tolerance and the tolerance drops because of the sense. So the leaf tolerance is not actually uniform across the plant. So, top leaves which are actually functionally very important, but it clearly shows that they are not really very tolerant. Now, because it is a workshop and you people probably have been, learning many things, not just fundamental and applied biology, but also different kind of, equipment and sensor and gadgets. So when I am showing you all this, beautiful data and understanding scientific finding, you must be, thinking that what all techniques one uses to study all those things. So in the crop science realm, in the plant biology realm, in the ecology realm, we use different kind of sensors. And I will quickly tell you some of the instruments which we really use a lot in the lab. Like when we measure photosynthesis, we use this infrared gas exchange analyzers. Now, different companies they produce. So, I hope you're understanding that I'm doing a little bit of gear shift. I'm trying to give you some quick idea about all the sensors that we use to do all this kind of measurements which I have been showing you. So this is one of the equipment. Its technical name is infrared gas exchange analyzer, but it has different names manufactured by different companies. This is a LI600, 6800. And you can see I have different, like at a time, I think I have been using 3, 4 LI6800 on different kind of crops I have been measuring. And this was one of the workshops where I myself was attending. It was really great. And this is another infrared gas analyzer, which is not from Leica. This is from LC Pro. This is also another kind of infrared gas analyzer. Probably it is from Walls. This is another kind of infrared gas analyzer. This is for survey measured, for quick measured. If you have 1000 plants, if you want to measure them over 1 or 2 days, then you need something very smart. And this is particularly meant for that. Now, many of these instruments work on the principles of chlorophyll fluorescence. Fluorescence is also something which we also detect to understand how our crops and plants are responding. So probably, I think some of you already know that probably you people measure fluorescence of the plants. And the fluorescence is the energy which the plant is actually giving back to the environment. So that's why receive and return. Receive means the light which plant is receiving. Return means the light which plant is returning back. But we can't see this light. We can't see this fluorescence returning back because it is going at a infrared region. Okay, and we human eyes are not visible. We cannot we can visualize the infrared. But there are instruments like different kind of very high throughput, high precision fluorimeters, which we have been using in the labs, and I'm sure many of you probably have been using in agricultural and crop physiology sectors. Different kind of fluorimeters are there. This one is very interesting. This is a continuous monitoring fluorimeter. This will be continuously monitoring. But there have been many sensors in the market. This is not a fluorimeter, this is a thermal sensor. So very smart experiments, you need not to have something very expensive, very costly. Even with the simplest equipment also, if you really understand the topic, you really understand what you're really looking for, and you have a very good idea about your model crop system, you should be able to really, address research questions using very simple tools. Of course, you can collaborate with other labs where they have more higher capacity. Now, look this, you people, many of you are from the North India, and North India, we know already the winter probably have started. Unlike in Kerala, where we are in Kerala, I am in Kerala, the winter is not here. Here seasonality is not much. Now, this plot, as you can see, it is showing the photosynthetic, you know, optimal level in the cool season versus warm season crop. Now, the cool season crop, if you see brassica, wheat, barley, their photosynthetic maximum capacity is between 20 to 5. Crops like sorghum, millet, maize, their photosynthetic peak is around 30 to 35. After that, it will drop. So now the question is, if the wind, when cool, the solar what will be the fate of our Indian winter crops? For an example, you take the example of mustard. Mustard, all of us we know, we have a huge rapeseed mustard belt in the northern and the eastern part and the western part of India. And mustard is predominantly a winter crop. Now, what is the fate of this mustard in a global warming scenario? Mustard is very interesting because not just the leaf, its green silic, those are also doing photosynthesis. But mustard has very important challenges. The yield is unpredictable under climate warming. Very limited knowledge whether this mustard, this kind of winter plant can acclimate, adjust to the warming, because the winters are becoming shorter and nighttime warming is happening. Abrupt temperature is rising. Someday it is suddenly hot. Because of all these things, uncertainty, and also it is compounded by the water scarcity. There are reports that as farmers have dropped suddenly mustard because they cannot give water to the mustard, because mustard is a very wide, a water demanding plant. So the question is whether these winter crops can adjust, they can acclimate, their photosynthesis can acclimate. If it acclimate, then this blue line, which is the photosynthetic performance, this optimal point, this circle, that will shift towards the left, sorry, towards the right, which means it is acclimating. When it cannot shift here, you see the crosses, which means it's not acclimating. We really don't understand how our crops, Indian winter crops like mustard, rapeseeds, and so on, they are going to behave if there is a mean temperature rise, or even when they are growing at a late season or a very late season. So this kind of things we really need to understand. Some of the experimental data, if you see, when you look into this mustard performance across a diurnal pattern, like from morning to evening, you see some of their photosynthetic performances, their photochemical performances, they actually dropping in the midday. But some of the genotypes, they performance, they do not drop. They maintain, there's a moderate drop, but not a huge big drop. But say many genotypes have a huge drop. Why there's a huge drop? Because that is the time when there is a more heat in the ambient. In the midday, there's a more high temperature. And as a response to that, the performance is dropping. So already, if you take a good notice, you will see that there are natural genetic variation in the mustard crop. This is just an example where you have certain genotypes which are relatively resilient to high temperature. So these genotypes we need to identify, we need to work on. Now I will slightly do another gear shift. We have been talking about crops, C4 crops, C3 crops. Mustard was a C3 crop. And now I will talk about a little bit of horticultural crop, because horticulture is a big deal now. Because when you go to the doctor, doctors are telling, okay, you have been eating a lot of rice and chapati and all. Better, little bit, cut it down, eat more fruits, eat more horticultural crops, nuts, fruits, all kind of vegetables. So horticulture is also a huge wealth of Indian agriculture. I will talk a few things about citrus. Citrus, we all know, we love citrus, right? And winter, because on purpose I chose this because winter is the season for citrus. We all like to eat, this orange, and it comes under the big family called citrus. Not just orange, even lemon also is under the citrus. Overall, we call it a citrus. And if you see, this is a very wonderful citrus garden, but this is not from India. This is in Florida. So 3 years back, I was in Florida and I was working on citrus extensively. I was working, but this is a very interesting scenario. This is a disease called HLB, Huang Longbing, or citrus greening. This is a very dangerous disease of citrus. I mean, if some of you don't know, please Google. Please Google. This disease is already there in India. And this is like a cancer. It's a systemic disease. It is caused by a phloem-limited bacteria. Okay, so called, the CLAS bacteria. Once it goes inside the phloem through this Asian citrus psyllid vector, through this small psyllid, when it sucks, it lands and it sucks. It actually introduces the pathogen inside the phloem, and the pathogen inside the phloem, it starts building the colony, it blocks the phloem. Now what happens? Its phloem pathway is blocked. This is the plants need a phloem, right? They do need to transport their sugars to the phloem. But if the transport vascular bundle is blocked, then how the hell plants are going to survive? So because of that, once this disease is there, the plants will never get cured. So people who know about this disease, very good. People who do not know, please Google. Now, because of this, what is happening that this disease won't kill the plant overnight. Healthy plants almost takes 10 to 15 years to show very bad symptoms, and eventually, it dies. But interestingly, we have been, traveling to we traveled many places in Florida, and also we moved from Florida to the northern part to the northern state, Alabama, which is just above the Florida, which is a more cooler, which is more temperate, where if you see, the citrus trees are growing under the canopy of this big pine and pecan trees, under the shade. This is under shade cultivation. Now, why under shade? Because the local farmers there, the growers, the citrus growers, they have a huge chilling time during the winter. The winter really is very chill. There is no snow, but still it is chill. And that time the citrus, they do not like this chill weather. But when they're grown under this big tree canopy, they are well protected. They survive better. Interestingly, under this shady condition, we see that there is no disease. These citrus plants, they don't catch this HLB disease. They are disease proof. How does this happen? Same observation we found it when we went to a natural reserve, bioreserve, which is a huge kind of bioreserve with lots of trees. And in between these big trees, we had this understory citrus trees called grapefruit trees. These understory trees were growing under cover of these big giant trees. And all these citrus trees, they don't have a disease. they are not having the HLB disease. Here I'm showing this area, circle B bar with this gray area. And if you see near that area, there are all the citrus fields are there. All these grooves are there, which are everywhere the plants are disease infected. But once if you look into the plants which are inside this reserve, under these big shades, those plants are not affected by the psyllid. Actually, psyllids did not come, did not land, and that's why the plants remained disease free. So shading, low light condition probably have something to do to make them disease free. And this is just a data, but when we did a survey of 60 under 20 trees of citrus trees, we could not find psyllids. And out of 60 trees, only 3 trees actually we found HLB infected. And even the infected trees also, they don't show any symptom. So we always this natural, finding, natural study, we have reported this in the Citrus magazine, monthly magazine. Also, if you look into this podcast, sorry, my voice is little down. If you look into this podcast, this this podcast will be talking a little bit more detail about this information. It is like a story. We have narrated this in the podcast. So from this experiment, outdoor observation survey, we got the clue. We did this shading experiment in the citrus garden in the experimental plot. We grew the citrus trees under different kind of shaded condition. We created artificial shade. And under different shade condition, as you can see, 30, 70, 50, and 0 shade, we observed these plants for a couple of months, and then the study continued for almost like 3 to 4 years. Now what we found is, and remember all these trees are infected trees. They were growing almost like 3 to 4 years old trees. On the top of them, we created these shades. And we have continued this experiment for next 3 to 4 years. And after that, what we found that this 30% shading is actually increasing the canopy volume and all the canopy growth parameters much better than under full sunlight without any shading and under 70% shading, which is like strongly shaded condition. So shading is actually protecting this infected plants, this HLB infected citrus plants. Now the question is how? Okay, before we go to how, this is the fruit, this is the yield data. If you see 30% shading here from 2019 to 2021, these are the parameters. These are the years when we were collecting this fruit yield per tree, fruit number, fruit weight, and some of the juice characteristic like Brix and Brix by TA ratio and some other parameters. So overall, just to summarize, your 30% shading, as I was telling you, that it has actually improved the plant productivity and the yield. What is the physiology behind? If you look into this particular response of stomatal conductance, like the stomatal opening in the citrus, look into this light blue color and this dark blue color, these two blue colors, which are the 30 and 50%. They have a better stomatal conductance. They maintain the shading 30 and 50, they maintained a better shading. So they maintained a better stomatal conductance. So the plants, with these infected plants, they were able to open their stomata, fix more carbon, which has contributed to their higher yield. In comparison to this 0 shade, which had no shade, and the extreme shade, which is 70%. So moderate shading or optimal shading was really a big player in boosting the yield of these infected plants. Also, if you look into this different seasonal pattern of their photosynthetic efficiency, again, we have seen that shading improves the photosynthetic performance, particularly in August month, the month where there is a very high heat and temperature in Florida, in southern Florida and central Florida. So under heat, citrus plants were really not doing good, particularly this infected plant, but shading was really good. As I see some cartoons, filling less trays in spite of HLB. So shading had really given a lot of improvement. Optimal shading, you have to remember optimal shading, and some of this we have reported in different articles and magazines. And in the podcast also, we have given some of this information. Citrus also, like sorghum, it is a megatherm plant. If you look into this citrus photosynthetic performance, here, sorry, leaf temperature performance during the midday, look into this, your air temperature and leaf temperature. Air temperature is this green line, whereas all the colored lines are leaf temperature. If you see during midday time, the leaf temperature can grow much above the air temperature. So citrus is also a very interesting plant which shows a megathermy response, which means the leaf temperature will go much above the air temperature. So to have a good citrus, taking care of their leaf temperature is very important, and also equally important to know if there are already genetically some varieties are available which have a higher heat tolerance. And if you look into this publication and some of this data I am showing here citrus has a very interesting heat tolerance range, ranging from low to high. In terms of Fahrenheit, it's given because America, people are more familiar with the Fahrenheit. So I constructed this, but in this publication, it is given in degrees Celsius. Please take a look into that. Almost, around 22 genotypes I have studied. Almost 2.5 degrees Celsius range is there, which is a pretty good range for a horticultural tree species, which is evergreen. And people who have little bit more idea about this evergreen, lot of these fruit trees are evergreen, horticultural fruit crops, citrus, lychee, mango, sapota, they are all evergreen trees. Evergreen trees by nature, they're a bit conservative. They don't like to change much. Their physiological property, which is evolutionarily constructed since a long time. Yet, it will be very important to find the natural variation in certain tolerant characters like heat tolerance. So some of this have been also have published in industry because for the stakeholders. This is an interesting treatment where we used kaolin K spray on the citrus in the peak summer month. This clay particle, when we sprayed on the plants, it looks different because we used red and white kaolin. It actually gave very interesting results. It actually deters the insect landing, and also it created a certain kind of micro environment on the leaf where the leaves really started performing better. These were all again infected trees. You have the goal was to how to keep this infected tree run for a long time and don't suddenly collapse and drop. Because these are long-term investment for the farmers. Once the citrus is planted, at least these trees the farmers expect it will, grow for 20-25 years. Now, if it catch the infection, it will die, and the productivity will come down. So a lot of this package and practices I was a part of, and even now also have working. This is a publication, and knowledge sharing with the stakeholders, with the growers. This was very important because whatever I work and I worked in the past, working with stakeholders, sharing knowledge with them, and also, like learning from their experiences. That is very important. So all these things should go together. Then only, we understand as a scientist that what is the research need, which is ground reality, and what is the research need, how we can help, whether through fundamental biology, through translational biology, or through hardcore applied research. So it is very important to keep for a scientist to keep him or herself updated with the ground reality, and also to communicate with the growers, the farmers, what kind of information you have in your lab, which they can make use of. So I will end with that right here. Thank you very much. And this is my email ID. You can always contact me if you have any other questions. But yeah, feel free to ask anything. And I will stop right here. Thank you very much.
Nandini.
If anyone is having any kind of questions, you can either type or you can directly talk to sir. The chat box is open now. You can type your questions.
A lot of thank you message. I'm also very happy. But yeah, I mean, if you have any comments, please feel free to reach out to me, email me, and we can always discuss.
So you can share your mail so that everyone or
Yeah,
anyone who is interested can directly contact you.
yes. Also, we have different kind of internship and summer school winter school programs here as well, but not that many seats are available. So, but if somebody is interested to, attend for some kind of small internship or some kind of winter school classes, they probably can also contact the School of Biology and so on. And also some of the research publications, what I have, it's already there in my website. If you go to my website, you can go to through either link. That is also possible. A lot of publications will be there, which the workshop trainees they can make use of this. Yes. So one question is there any correlation between shading and pathogen infection? This is asked by Dr. Avinash. So what happened is like, yeah. So the experiments which we did, the field experiment which we did on the citrus trees, the trees were already shaded. So sorry, the trees were already infected, and on the infected trees we gave the shade. So what happened is the plants were already infected by the pathogen, but what we observed that the disease progression or the symptom progression was affected, not the infection itself, because they were already infected. But after infection also, at a certain speed, the disease symptoms will progress. The optimal shaded plants like 30 and 50%, there the disease progression or symptom progression was much slower compared to the plants which was not shaded, like fully open, zero shading. So disease progression wise, there was a difference we saw.
Sir, we have one more question.
Okay.
Is there in the one of your slides where you
Huh.
used shading in citrus, it was a black shade material. Is there any
Huh.
difference when you use green types also?
So Green, so I personally did not use a green screen, but there are growers where growers means citrus farmers where they have used the white and the red screen. So green screen probably nobody has used, but white and red screens people have been using there, which has an effect on the citrus growth. Definitely. And what kind of effect? Well, it effect is not same. There are different effects. For an example, red shading is shown to improve the citrus water use efficiency, which I have seen. White shading has improved the light, what I can say, the light reflectance better inside the canopy. So they have a better light regime inside. But overall, any screen you take, the major goal was to avoid the infection spread, sealed landing. Top of it, the shading color, like whether it is a white or black or red, that will subjectively contribute to the quality. But to the change in the tree, which is not same for all. It is slightly different. Any toxic effect on pathogens? Not really sure what you meant by toxic effect on pathogen. Uh, you mean the shading effect on the pathogen? If that is the question, then yes, there is sometime we have seen that because of the shading, sometime the pathogen level, which is a PCR based assay, we have seen that sometimes the pathogen level drops inside the tissue when we, take when we do the PCR analysis of the SAP to understand the pathogen titer level, we see that the pathogen ratio drops, but it is not very constant throughout the year. Sometimes we have seen that the shaded plants have a very less pathogen level compared to the non-shaded plant. White particle film cost. So cost wise, you have to check. I better not say anything about that. Check and figure out. White and red alternatively can we use Sarji? Acha, white and red alternatively. The thing is, you have to check which works in your scenario better. Because you see, of course, probably others, Dr. Varsha is that this is actually a good question. This kaolin clay is basically a clay. It will wash out with the rain or if you do some kind of foliar spray. So first thing is we have to use some kind of adjuvant so that the clay stay back on the leaf for a long time. And I think the red, if you look into the publication, I think the red kaolin spray has given better results compared to the white. Now, whether red and white alternatively can be used, though that is an experimental question, research question. But of course, a farmer or a grower can try it at a small scale and then check which one, if there is any treatment effect on that.
Session was too good, sir, and I think it was self-explanatory as well. So if anyone is having any question, you can directly talk to sir also instead of
Sure.
typing. Okay, there are no questions. So we'll conclude the session. So thank you so much, sir, for your presence and your deeply insightful and thought-provoking session today. Your lecture beautifully connected plant physiology, hydraulics, climate resilience, and ecosystem sustainability in a way that was both scientifically rich and very easy to understand. Your clarity, your simplicity in communicating complex concepts, and your passion for plant science have made today's session extremely meaningful. It has opened new ways for us to approach research, especially in the context of climate change and resilient agriculture. On the behalf of entire Winter School Bharat 2025 team, we express our heartfelt gratitude for your valuable time, your enriching knowledge, and, and your warm interaction with all of us. Thank you so much, sir, for making day 27 truly special. We hope to have the privilege from learning from you again in the future academic activities.
Okay.
Thank you so
Thank
much,
you.
sir, for your
Thank
presence.
you.
Thank you,
Everybody,
Dr. Saab. Thank you,
Thank you.
Dr. Saab.
Thank you. Thank you. Thank you.